Merci beaucoup. And you'll be thankful that will be my last 2 words in French that I will utter. Although I do have a wonderful story about walking around Paris in 1983 with Mike Phelps, in which I was the resident French expert, and how we almost got into some very serious trouble with some ladies of the evening, but that's another story. So I also want to certainly thank Roger for the invitation to come up here. It's great to come to a wonderful, cohesive group who's doing PET research and clinical in the right way. I do feel a little awkward because, as I think Roger mentioned, we had gone to many conferences and gave presentations at seminars, and I was always right after Roger. So I'm just feeling a little uncomfortable that he didn't give the first talk. So I will muddle through somehow as best we can. So what I'm going to do is talk to you today about a range of applications of PET in neuroscience. I know that's not the big focus here, but a lot of the ideas that we use there, we're trying to move to other parts of the body. And in many ways, I think that we had certain visions how we set up doing brain PET a long time ago, in no small part influenced by the key work of Lou Sokolov, who just recently passed away, who invented 2DG, which led to FDG. So what I'm going to try to do here is give you a little overview of research PET that you don't really need, but I'm going to do it anyway. And then talk about a number of different projects. And I'm just going to be going through them a little bit quickly. Please interrupt with questions about different ranges of topics in the development of characterizing a tracer, pushing some receptor pharmacology a little bit further, looking at novel targets and taking them into the clinic, novel applications, imaging neuroinflammation in the human brain, and then finally, an area we're very excited about is a brand new target that we think will let us measure neuronal density. I don't need to show you this slide, what makes PET cool, why we're still doing it after I'm 36 years doing PET now. That it is, we can be quantitative with it, we can be using biologically relevant molecules with the isotopes that we have. We can be quantitative, we can be translational, key to do the animal studies paired with the human studies, and we can apply tracer kinetic principles to turn radioactivity images into physiology. But here's where the money is. Thank your chemists. Take a chemist to lunch, okay? That's a good thing because this is having a wide range of tools. That's the list of molecules that we have currently implemented there. I think we're about 43 into humans at this point and over 100 in animals there with a wide range of topics. But the ability to look at many, many different targets and be able to do the novel chemistry and put that all together. And a few places forget about that. A few places think that PET equals FDG. And we know better that that's in the case. But also to be able to do that quantitative, to take our images and watch how they change. Here's some images of a serotonin transporter ligand in the brain, early, middle, and late post-injection, and recognize how the images change. And most of you know that, and then how do we decide how to analyze that? And that's where we bring in the tools of kinetic modeling. So I'll belabor this briefly, just some of that background on what do we do, where we can take the dynamic data and incorporate some model to characterize the time course of the uptake and the distribution, usually making measurement of the input curve, the time activity curve in the blood, or a surrogate for that, with the goal to take radioactivity and turn it into physiology, quantitative physiology. How do we do it? We start with boxes and arrows, and then we try to describe what might happen. So we might have a ligand binding to receptors, binding non-specifically. Then we make it simpler. We recognize that although we have beautiful state-of-the-art scanners, we don't have a PET MRS, because that would be cool. Because if we could tell the chemical form of our molecule at the same time, that would be fantastic. Then we could separate these boxes. Instead, they're one big lump. So we simplify them. We come up with simpler models. The goal here is to describe the time activity curve in a way that has biological and clinical relevance. Something that can mathematically let us convert what we can do in vivo to what we would do if we were taking out tissue and making an in vitro measurement. And we've developed some terms that we've promulgated. Some of these terms, the volume of distribution and the binding potential. I apologize for these terms. They're really not very good. But we've used them a lot in the literature, and it's just something to get comfortable with. The volume of distribution is a ratio. It's a partition coefficient. If you took chemistry, you'd never call it a volume of distribution. If you come from physiology, it's kind of a volume. It's the volume of blood that would hold as much radioactivity as 1 ml of tissue at equilibrium. That's not very useful. Binding potential is a ratio, and it's a ratio that's got a bunch of stuff in it. We play with that a lot, but it is getting more directly related to the available receptor, we call the avail, and the affinity of KD. Now I know some of you are MR folks, and I usually belabor and have nice, pleasant conversation with my MR folks because we claim to understand what we're measuring. but claim many quotes and things like that, because we try to dive in to explain what it is we're measuring and try to make biological inferences there. In MR, there's a lot of great signals, and there's a lot of great people trying to do that physiology and try to understand that very, very well, and it's a little bit harder. Some of our tools are a little bit more effective for them. So how do we do this analysis? And there's a number of different ways we might do it. We might do it the hard way, where we collect these time courses, things you do a lot. We collect time courses in different regions. We would usually measure an input function somehow. In large animals and humans, we can do that. In mice or in small animals, we might need a surrogate. We're going to describe these curves with kinetic models and extract parameters. If we're good, we can do that very, very fast. We can find rapid estimates. Every pixel of the image, we can create a parametric image. So here's an example when I looked at those dynamic images for this particular tracer, this Prozac analog that shows the early distribution. Let's try that again. Good, early distribution, changing to a more receptor distribution. If we take every pixel, generate its time course, estimate a model, we can separate it into delivery, or blood flow related information, and VT, this volume of distribution, or receptor related information. Why do we do that? Do we need to? We've got a lot of pretty receptor related information here in that late image, and clinically that might be what we need to do. But when we're early on evaluating something, we'd like to do the science well enough to properly separate those out and be quantitative. It also works when I put on my engineer's hat and say, how do I want to make best use out of this data? Or when I put on my ethicist hat and say, how little can I inject in a human being to get a good quality result? We do a lot of other techniques to try to make our measurements better. We do lots of measurements with reference regions. Instead of using blood, do we find a surrogate region that we can use to be able to make a measurement? Recognizing that, thankfully, by doing IV injections, for most of us, the blood delivery to the organ is very similar throughout all the objects. And we can use that surrogate to infer what the input function would be. This is fun from those of you who have fond memories of taking calculus, because we can actually use integration by parts to solve the equation of this one. It's the only time I've used it in 35 years. But when you do it, you can actually use that pixel by pixel to create images, some old images from Roger Gunn from Raclopride, to get a reference region, to get these quantitative measurements without the arterial input function. But you know what? Even that math is a little too scary to people. I won't ask for you to name names. So it's better if we could do it even simpler, at least for some people there. And so we've come up with these graphical analysis approaches. And these are just nice approximation approaches that get at the meat of what we're after. It's actually very powerful because they recognize what is the data trying to tell us. And what was found was by rearranging these equations of the tissue activity and the blood activity, you can get it into a straight line, like that one. So here's one where we see 3 different regions with a serotonin 1A ligand, where the higher the slope, the more the specific binding. People like these graphs because straight lines give you a warm, fuzzy feeling about your data. It's really hard to get when you have time courses like that. Very few fuzzy feelings from curves like that. But these are great. You know that you have a straight line, it's telling you something, and you can measure it, even with something like Microsoft Excel, one of the great sophisticated PET analysis tools that are out there. But even this was too complicated. Wouldn't it be nice if we didn't have to even do that at all? Maybe we need to make the brain look like the in vitro dish by coming to equilibrium. And in some cases we can do that. We can infuse the tracer. And this is where we use our engineering in a different way. Instead of a bolus, we give a constant infusion. We try to get it to equilibrium everywhere. And then those binding potentials just jump out of the data. Especially when our data then look like that. It's very easy to read out a flat value. We don't even have to figure out a slope. So lots of different math approaches. This is good. This is job security for the modelers, that there's always a different way of approaching that data. On the other hand, it also makes the literature pretty confusing about one method or another. Hopefully no important results really depend on the subtleties of one method. What do we do with these tools? Well, one place we do them is in drug development. At Yale, we do a lot of work with the pharmaceutical industry. We've done studies with, I think, 15 different companies. And, there's a lot of different approaches. They're interested in the drugs to prove, does it engage the target? Or does it engage the mechanism? Or will it be efficacious? But what they really want is a slide like this. This is, I call, my vice president slide. Because this is an example of a study where we've done, in this case, histamine 3. We took a subject and gave a baseline scan. We gave them the drug that also hit H3. And the scan looked like we wiped out the brain. And then, of course, we brought the same subject back the next day, 24 hours. Now the drug is clearing and the receptors are coming back. I call it a vice president slide because they can understand it. Okay, it's very clear, and they clearly know their drug hit the target. Now that doesn't stop us from having to do all of our quantitative science, how we will use our registrations to atlases, and generate our time activity curves, and fit them to models, and calculate parametric images, and estimate binding potential values, and forgive me for going fast, but then to do these occupancy calculations so we can get this slide. This is the money slide here for what we do in occupancy plot. This is where we have studied a drug at different blood levels, different PK levels, versus the PET measured occupancy throughout the brain as visualized by those images. And we can get, in this case, a very nice, clear, not just dose-dependent, but blood level-dependent response. Now, my undergraduates, when I would introduce this, I would ask them the question, why would a snobby US Ivy League university like Yale want to bother to collaborate with those people from the dark side in the pharmaceutical industry? And being all these active Yale undergraduates, they'd all raise their hand and say, money, okay? And they'd be right, but what's really cool is when we do collaborate and it helps us either bring up a tracer we didn't have by funding an early study, or help us develop ones that's brand new. And I'll show you a few examples of what's coming next, of places where the collaboration with pharma has really led to a win-win. They've gotten an answer about their drug, that they need to have this blood level to get that occupancy level, and it's helped us build a new tool that we can use in clinical research. All right, so let me take you through a number of different ideas, different aspects of the kind of things we're doing here in brain PET, either in humans or preclinically in non-human primates. We're going to start with a target, a glutamate target called mGluR5, metabotropic glutamate receptor. Okay? So this is a very relevant, it's very widespread. Glutamate is the #1 neurotransmitter in your brain, the #1 excitatory neurotransmitter, and it's got a number of different subtypes of receptors, and the metabotropic ones are very relevant in the brain. And a while back, this tracer, we didn't develop this one, it's in the category as MTEP and others that have been used a long time in vitro called FPEP with fluorine-18, and we went about to try to use this molecule in humans to see what we could do with that there. And so this is originally, and this was a paper we published a couple of years ago, just trying to say, how do we want to do the study? Very simple kind of, you know, just kind of, which do we want to do? Do we want to do this in the case of a bolus injection or a constant infusion? Start with chemistry, of course. This is a nice compound with fluorine-18. We make it at good yields, forgive the millicuries there, with very good specific activity. And that's very nice from an operational viewpoint, because that lets me make one synthesis and study at least 2 patients, even back to back. Something to keep in mind when you give a pharmaceutically active agent that you want to work at tracer doses, if you wait a couple half-lives and inject the same radioactivity, you've just injected 2 to 4 times more mass. And you have to be sure you're working at tracer doses. So we did a number of subjects both with a bolus injection and tried to do the time activity curve doing it the hard way, and then that bolus infusion way. We were trying to reach equilibrium and hope that that would make our life easier and simple. And so we did a number of healthy controls in this initial study. We did them on the HRT, that's our high resolution scanner. We're pretty happy and lucky to have one of these. This is a 2 to 3 millimeter resolution, depending on quite how you choose to quantitate it. And it's our big workhorse for our brain studies, as you might imagine, when we can get very high resolution. It's also job security for the physicists, because there's certainly a lot of issues that even now are still resolving there. Now, when we first started in a research enterprise, we really weren't sure how long we wanted to scan. And so we took these scans way out there, going out, in fact, to 4 hours, having the subject get up and have a break. And part of that's to be able to register that data together is going to be very, very important. And we did the whole part. We did put in arterial catheters. We probably do about 300 of them every year. We take blood samples. We measure the metabolites in our HPLC lab. That's one of the busiest places in our PET center to determine the metabolite corrected fraction. Okay? And once we did some bolus studies, we could do some math to calculate how to do the infusion. Okay? And this is one where we have this term called K-bol, which basically says what's the ratio of the infusion rate, my bolus infusion, to the magnitude of the infusion rate, about 190. Another little trick we learned was this is a sticky compound. Probably our chemists knew this, and we just didn't communicate this well enough to us. So we have to use some special tubing that we use, PTFE tubing, so it wouldn't stick to the tubing, or our infusion would not end up being an infusion. We do everything we measure is in list mode. And in fact, we correct for motion by correcting every single event back to where it would have belonged if the subject didn't move. And then we register them to MR templates and generate the time activity curves. So here's some examples of images. I don't know how well you can see those there. One thing I did learn in California, and I like to say I went to grad school in Hollywood, and the one thing I did learn back then is if you have high resolution, show it in black and white. And FPET images are gorgeous, and on an HRT, they're spectacular. I mean, for those of us who go back a way, these are better than the earlier generation MR images there, just to see the spectacular involutions of the cortex that you can especially see on the coronal and the sagittal views there. Images visually look very, very similar depending on your time for either the bolus or the bolus infusion. Here's what the time activity curve. So things are looking very different. You do have clearance. Here we're going out to 6 hours. Here's a constant infusion, so you see we can, in fact, get to equilibrium. And then one of the tricks with equilibrium is when do you get there and when is it good enough? Are you there at 90 to 120? In most regions, some are. If you have a little bit of a trend, does that matter? Welcome to our wonderful world of not doing anything perfectly, but doing most things pretty well. Making a point where it's more important to be close and reliable than to be perfect. Perfect is expensive. All right, so we picked a time period, and we were hoping to be able to do it in a 2-hour time period, and so we picked that based on 90 to 120, and at this point, on average, we were certainly less than 5% mean value. Okay, so the first thing, we've just injected the tracer. We've analyzed it 2 ways. This is a tracer dose. We should get the same answer. The volume distribution, the magnitude, no matter which way we analyze it, it should be identically the same, and sure enough that we do. Here are the mean values in different regions of the brain. Some high binding in the caudate and cortical regions, lower in cerebellum, lowest in white matter. And this is showing for me the mean and the standard deviation across, different people. Where the bolus values looked like, and it turned out that on average they're a little bit more variable. The coefficient of variation was closer to 20%, and the bolus infusion we got closer to 10 to 12%. So that was something saying maybe the infusion would be valuable. And when you think about it, there's just less things to go wrong when you have to measure that blood curve and measure the metabolites and do all those corrections. You got to get the curve a little bit more correct. In the infusion, all you got to worry about is that you're flat, both in the blood and in the brain. We looked at time. We did whether we used 6 hours or 2 hours, and these are wonderful when you get slopes that close to 1. That says everything worked out very well. So our initial conclusion was that we could measure it well. The bolus infusion seemed good, seemed a little better, less intersubject variability, and we could do it in 2 hours. So now if we really want to be obsessive, and this one we end up being pretty obsessive, the next thing, the very exciting thing you do next is a test-retest study. Woo-hoo, okay? I mean, it's one of those things that's gotten very popular to do these things, and people say you can't publish something until you know the test-retest to know what's really different. I think it's pretty overblown. It does help you choose a better method, perhaps, in terms of the variability, so for that purpose it's good, but whether or not it's really going to be helping you, unless something is particularly bad. So we evaluated this one. This got published earlier this year, test-retest there, and basically we wanted to compare doing the full compartment analysis, even with the bolus, to the equilibrium analysis. One other important thing with equilibrium that would be great is, can we, instead of using arterial blood, can we use venous blood? If we're at equilibrium, we would hope that the artery and the vein would be the same. And when you think of it, this is the artery and this is the vein you're talking about here. There's not a lot of mGluR5 receptors in your hand, and so there ought to be a pretty good equilibrium there, although we've never scanned the hand, so I don't absolutely know that. So here's the example data set for a 2-hour scan, which is very, very nice. And what's really nice is not just that we have this nice flatness there, but the data quality are very, very good. This is a nice with fluorine, high uptake, and very, very reliable. We compared a lot of different methods, 2-tissue compartment modeling versus equilibrium against artery and venous, a graphical method called MA1, sorry for the bad production there. But what's also nice is we can then calculate these images very easily of equilibrium. And if we're at equilibrium, it's simple. It is the image just scaled by the input curve. Here's what it looks like from 90 to 120. Turns out we did it in color. We can even probably go a little bit earlier. This is one of those trade-offs of which is better. Have a shorter scan that might be a little wrong, or a longer scan that's a little bit more right. Not clear what the answer may depend on the patient populations that we're doing. Here's the important one. Does the artery equal the vein? And this, of course, meant we took samples from both. And we did metabolite analysis on both and did all that work. Not something you want to do regularly. Very happy to get that nice answer of a slope of 1.0. So then we also looked at that test-retest. We did 2 scans in each of these subjects on different days. And we looked at the variability here. They were all pretty good. And happily, the equilibrium won. That basically means our life got a little bit simpler, that we can just do a short scan. And the venous, we paid a little bit of price, but nothing significant there. So now our routine is that we will make a run of FPET. We'll inject our patients in the room outside, kind of like FDG. We'll bring them into the scanner at about somewhere in the 60 to 90 minute range, scan them for 30 to 60 minutes, take them off to a transmission scan, etc. at the end. And we can clearly easily do 2, and we've even done 3 patients in one day, back to back to back, all taking advantage of the high resolution scan. Okay? So this was this very simple-minded study, optimize the methods for a known existing tracer, one that has good characteristics, make it easy, make it effective, and get the best use out of your chemistry facilities too, to make one run being able to multiple subjects. And so shorter acquisitions are possible. So if we're going to take this into patient populations where that may be a problem, think about autistic spectrum disorder perhaps, a number of different areas where this might be relevant there. And the equilibrium method did seem to work very, very well. Okay, change of pace. That was very human, clinically applied, back and forth math, lots of slopes near 1. That was good news. Small variability is a pretty good story. Now we're going to push the envelope a little bit. We're going to do that in a month. Where we're going to try to understand what we usually don't bother to understand when we're doing a PET tracer. Trying to measure this in the KD. So if we're going to, the target we're going to use is looking at the transporter for glycine. So glycine is a co-transporter at the NMDA receptor, also glutamate receptor, and glycine affects and modulates the activities of this receptor. Now, a while back, a number of different compounds were developed because it was really interesting to try to come up with drugs that might block this site, for example, in schizophrenia, to be able to modulate some of the positive and negative symptoms that come with schizophrenia. And so, previous years, we had compared 2 different molecules, a Merck tracer, MK6577, versus a GSK tracer. Note these catchy names the drug companies come up with, 9311145. Internally, we give a name to every tracer just so we don't get too confused. We had been happier with the F18 tracer. And so our goal now is to say, well, let's see if we can learn a little bit more about that molecule. In fact, let's see if we can learn what the true tracer dose is. Hold that. All right, so the objectives here were to characterize it, but to measure this, to separate out Bmax and Kd. We talked about the thing we measured before is something called the binding potential, the available receptor divided by the affinity. What do we mean? Well, hopefully many of you, this graph will look familiar. This is a scattered plot. You've had some basic pharmacology. When you did any specific receptor target, you would start at a certain amount of binding. You'd slowly add more and more compound. You'd block more and more of the receptor. The bound would increase, and you could rearrange the data onto that straight line and be able to estimate that when you got out to the x-axis, that was the Bmax. The slope was -1/Kd. When we do a PET scan in a human being, where do we generally work? On this graph. It's always a good question to have people to figure out how comfortable they are with PET. And if you have a great chemist, you're working over here. You're working at very high specific activity, very little mass, very little binding. You're working way up on the y-axis. So what we're measuring is this y-intercept, which happens to be Bmax/Kd. That's why we measure it. It's the thing we call that binding potential, the maximum possible binding here, normalized by the Kd. And the vast majority of times we don't bother to try to separate them, to try to understand separately. So the dirty little secret in nearly every brain receptor study, when they say receptors are higher or lower or change in this disease, they really don't know. It's just this numbers change, and they're hoping all the other numbers don't change. And rarely do we change, because it's hard. Usually it means we have to give a pharmacological dose, and either that's illegal or it's unsafe or both. Now, in the past, people spent a lot of time trying to separate this out. It kind of fell out of fashion. It goes back to the 1980s. That's the last millennium for those of you born. Is anybody? Never mind. Lars Farda had some really early work with science doing it with raclopride, which was safe to give, and where he was able to give pharmacological doses and using classic receptor measurements able to estimate Bmax and Kd. Or the folks, Jacques Delforge and others, who formulated this very, very complicated multi-infusion one, try to estimate all of the terms with 3 injections back to back to back, and even this was done in humans in some way. Jim Holden from Wisconsin, in collaboration with the Triumph group, came up with a way of doing this with infusions, where basically you did multiple infusions at different specific activities, you partially saturate, you walk your way down that scatter plot to separate Bmax and Kd. For raclopride, that required multiple injections. We wanted to take that one further. We had F18, let's see how much we can get with one study. So this was done in monkeys, where this was our design, where we were going to give one very long infusion, to keep the hot at equilibrium, and then we're going to add some cold to partially block, and add some more cold to partially block. If life is good, this is the curve we're going to get. We're going to get to our first equilibrium, we're going to get part of the cold, get to a new equilibrium and a partial saturation, and more saturation. And one of the challenges that we needed to do with this particular compound, this Merck compound, was that unlike raclopride, there is no reference region. There's no region in the brain without some of these glycine transporters. So now I need to estimate the Bmax, the Kd, and the background, which we call this VND, this non-displaceable volume of distribution. Okay, so we did, looks like that didn't come out just right. So we did 2 animals. Sorry, it looks like something's weird on the slide. We did 2 baboons, and we did them at 3 different equilibrium positions. And certainly first, here's the images that we got. So this is various images from a glycine transporter. That particular one, there's a lot in the pons, there's a lot in the cerebellum, relatively little in the cortex, and even less in the white matter. Here's a high specific activity tracer level. Here's the blocking dose showing where we are accumulating. So then we looked at our time activity curves. Again, remember, 1 long hot, a medium block, and a high block, all in one day, 5-hour scan. This is a good day when you get your curve like that. When it comes out and you get exactly the down step as the math predicted. It means you have some combination of brilliant mathematical intuition or a hell of a lot of dumb luck. So I don't know what fraction I can attribute one way or the other, but it certainly worked out very, very nicely. So this gave us an opportunity to analyze this to death, okay? Because we could analyze this with regions of interest. That is, we could go back to the images and draw the regions and quantitate all the values. We could actually do this for every pixel. Every point, just like we did with the human images. So we did a version there where we did this for every pixel in the brain. This became a big quantitative nightmare. So when we had to do this analysis, this was our model. We were at equilibrium. We wanted to measure Bmax. We wanted to measure the affinity. We knew the cold. We knew the cold because we knew the hot. And we had to estimate 3 parameters there in green. We wanted to do that in a number of different ways. But we ended up with trying to estimate about, from 10 million measurements, about 1 million unknowns. Child's play. Okay. And so we did that in a variety of different ways. Here are our values when we did regions of interest results first, in baboon 1 and baboon 2. And the KDs we got were between 1 and 2 nanomolar. Remarkably, this agreed very, very well with the in vitro value. Sometimes that works, sometimes it doesn't. Sometimes the in vivo molecular system, the pH, whatever, is going to change the affinity. In this case, we didn't see that kind of a big change. And we were able to measure some Bmax values. Unfortunately, there wasn't a really good gold standard out there. It seemed to agree reasonably well with the values that we were seeing from human tissue, 30 to 60 picomolar, nanomolar there. We also looked at this where we pooled it in different ways, where we analyzed different combinations. In particular, we wondered, well, is that non-specific binding uniform? One of the other dirty little secrets we tend to do is to assume that the non-specific uptake of all of our molecules is uniform throughout the brain. We never check. We assume it. Occasionally we check. So we decided to check. Here's a case since we're estimating. If you remember, we go back to the equation, I can decide, well, maybe I can fix some of these or let them all float or some combination. So we estimated the receptors and the non-specific. And that let us produce, here's the numbers for this. We saw some variation, but ultimately we could produce an image. So this is an image where pixel by pixel we've estimated Bmax, the receptor concentration, as well as the non-displacement. And it's got some variation. The good news is it has variation in a reasonable way. It's higher in the white matter. And that kind of makes some sense. A lot of our molecules, they're very lipophilic. The white matter, thanks to myelin, is more lipophilic. You'll tend to get just higher partitioning, higher non-specific partitioning. So it's just one of those messages that says you need to be a little careful if we're going to use white matter values as an estimate of non-displaceable in the gray matter. But it worked out pretty well. So we learned a lot, and it got published even better. And we could learn how to understand it. We learned that what the in vivo affinity of this was. We learned that we were very, very, very safe in our dose. We were nowhere near a tracer limit. If we know the KD, we know what would occupy anything at a pharmacological dose, and we know that at the specific activities we have, maybe we're occupying 1 or 2%. I think it's very, very good. We got pretty good agreement, which was nice. Doesn't always work. And we've progressed on to human studies there, as shown in that making your head spin version over there. New topic. And I'm sorry, bouncing, but just kind of give you a feel for yours. The process of hitting a new target in the brain. And this is one of those where we did collaborate with the pharmaceutical companies looking at kappa receptors. And I know there's some interest here in trying to look in the opioid system. So there's three kinds of target receptors in the opioid system, mu, kappa, and delta. There's been mu and delta out there for a long, long time, mu over 30 years, and there hadn't really been selective kappa receptors. And there's a lot of reasons and a lot of diseases, pain, depression, alcohol dependence, etc., where kappa would be interesting. Okay? So the compound that we're working with is this other, also one with a catchy name, LY2795050. For those of you who speak pharma, the LY stands for Lilly. And that's who we work with on this collaboration. And that worked out very, very nicely. And when you think of it, they have these libraries of molecules they're looking to develop as a selective agent. They've chosen one because it might make a good drug. And then they can help us pick target, or at least the basic backbone that's going to make a good tracer. You know, as a general rule, a good drug is a bad tracer, and vice versa. A good drug, what do you want? You want it to hang around a long time, take its time. So that way you only have to take it once a day. We want it in and out. We want it fast. So usually it's going to be a bad drug. That's good, so there's a little less competition that time. This one's pretty good. The affinities, we had an affinity about 1.37 nanomolar. Big selectivity over mu, 60-fold in vitro. The chemistry was complicated. We used cyanide, and hopefully no one will ask me questions about this slide. But we're able to produce the compound. The specific activity is a bit of a challenge with this molecule. And this has led us to try to be looking for better ones there because of the nature of the cyanide chemistry. It's important whenever you're hitting one of these targets. We took this into healthy subjects after we did a lot of monkey work, again on the HRT. What's nice here is you've got legal drugs to give blocking. We use naltrexone, which has been readily used for a long time. It's not a selective agent, but it lets us hit and specifically block the target there. And so we're doing baseline scans and blocking scans, 150 milligrams of naltrexone there. Lots of different modeling methods, lots of different outcome measures, and also a challenge on that block, that non-displaceable. What's nice with a blocking study, you can validate, is there a region with no kappa binding? We didn't find any. Seems to be pretty ubiquitous in the brain. So again, we're going to be stuck with how do we estimate the non-specific, how do we subtract from that. So one thing we do is we make the measurements in the blood. This is the total activity in blood. This is the parent fraction. That's from the HPLC measurements. You see baseline and blocking. This is nice. The blocking didn't change it. And we see that that's the mean and standard deviation across our healthy controls. And the final input curves we use for the modeling. Here's what the images look like. Again, on an HRT registered to the MR. We have high uptake in the amygdala. You see it in the insula. And you're seeing a bit into the anterior cingulate. When you block, it wipes things out very, very nicely. We do our time activity curves, and what we've done here is you're just breaking the regions up with different analyses, different fitting methods. We compared 1 and 2 tissue compartment models and our linearized methods MA1, and the control data, and the blocking data. This is one of those questions when you look at these time activity curves, you know, this is where the money is down here, the separation between the different regions. And when you display it on this full scale, it looks small, but really what matters is the ratios that you're getting down here. These are not amazing ratios, these are okay. It's enough to be able to do some useful things. So we measure the distribution volumes, this equilibrium ratio that I alluded to before, values up to the 3s in the target regions, in regions with what we thought was not supposed to be much or any blocking. Kappa was in the cerebellum. When we blocked, it still goes down. And so we saw everywhere, not a lot. So the cerebellum is close, but it is not an ideal reference region. Another way to look at this is what's called an occupancy plot. This was a rearrangement of a plot that Niels Lawson published in the early 90s that Ben Cunningham and Roger Gunn did, where he rearranged the change in blockade against the original values. The slope gives you the occupancy, in this case 98%, and the x-intercept gives you this non-displaceable, a number that's a little bit lower than our cerebellum numbers, about 1.95. So we found that our cerebellum is close, but not quite right. So we have to come up with a way of doing it. How do we do it? We said, well, let's propose this. We don't know if this is fair. We're going to say we'd like to have something that doesn't need blood measurements. Perhaps the cerebellum will be useful, but we have to fix it, in this case with a fudge factor. That assumes, in our particular disease, that there's no specific effects going on in the cerebellum. Do we know that? No, not unless we do our homework with the blood curve. So it may be useful, but we're not sure if that would be the case. And of course, after we did the modeling and we figured out how to analyze it, the next exciting thing we did, of course, was a test-retest study. Sorry, these are getting pretty boring, but again, you want to find out how good things are. Again, different modeling methods, different normalizations. Here's one point that I wanted to make here was that we measure that distribution volume. that ratio. One thing that controls that is how much protein binding you have. The more protein binding, the less is going to get into the brain. We call that FP, the free fraction in plasma. And so for some tracers, it's better to correct for that, especially if you can measure it reliably and if it varies in different patients or at different times. So we're going to look at that measure or these binding potentials using the cerebellum. And we look at various measures of the test-retest variability by comparing the test and retest values. If you read the literature, people do it in different ways. Some people do it without putting an absolute value. Some people put it with an absolute value. They have different ideas of what we're looking at there, so we do it both ways. Here's what images look like here. We've done average images across many, many subjects, test and retest. Get a little blurrier when you do that. That's why they're in color. And then we looked at the test-retest reliability in different units. I'm going to focus on the absolute value. It's the simplest way to do that. And we're certainly getting, in terms of our volumes distribution, the total binding, very good numbers, 7 to 8%. If we correct for that free fraction, things get worse. And that's because this number is very, very low. It's under 1%. It's hard to measure that well. This is an in vitro measurement made. We do it with ultrafiltration because it has to be fast, and it's hard to do well. So this was a case where, well, it could be in a given patient population, we can't measure it reliably. We're not going to be able to correct if there is a true difference in protein binding from one disease to the next. So this is pretty good, but this isn't really the measure. This is the total binding. We're really interested in the binding, the specific binding potential. There the variability goes up a bit. And this is one that's very common on the regions that have high binding. So in the insulin, the anterior cingulate, you can do pretty well. As you get less and less binding, your percentage error gets higher just because the percentage of signal that's specific gets lower and lower. So we've gone pretty far with this one. We figured out how to do the analysis. We're pretty comfortable. We have a nice selective and specific signal. We have decent test-retest, very good on volumes of distribution. And our binding potentials are okay. They're not to write home about. I mean, we're having values on the order, I don't even have the numbers here, on the order of 1 to 1.5 in the highest binding regions. So it's not great. And what does that mean? That's a grant opportunity for your chemists to make something better. And so Henry Wang, who's our lead chemist, has a couple different grants on developing novel agents for kappa, both for antagonists, like this one, and agonist tracers. And 2 of them are already in humans and will be, the early evaluations of some of those were shown at the Society of Nuclear Medicine. Different direction. What have we talked about so far? We've talked about basic modeling analysis with a tracer. We've talked about a little bit more in-depth analysis where we're going to give pharmacological doses to an animal to take apart something we don't usually do in humans. Brand new targets looking at kappa. And we've now begun to use this in PTSD and in depression and a number of different disorders. Now we're going to look at trying to image neuroinflammation. Neuroinflammation is high. Every disease seems to be related to, associated with, indicative of neuroinflammation, if you read the literature. This is hard literature to read. Actually, this is an interesting one for those of us who start as imagers, is we have to be taught, we talk to everybody, right? You know, we're trying to do beta cells in the pancreas, and we're trying to measure effects in tumors and changes, and we're trying to look at small regions and activation in the brain, and which means we have to learn about the pharmacology and physiology, and boy, is this one challenging. If any of you are reading this literature, understanding it, how it's evolving quickly, it's challenging. So, as we said, it's high, and it's relevant in you name it. Clearly, it's something obvious like multiple sclerosis, but even in Alzheimer's disease, in psychiatric disorders, everybody says neuroinflammation is relevant. How does the brain work? What's the brain's immune system? It has a dedicated system called with microglia. How do they work? And I'm going to get, if anybody's an expert here, I'm going to get on dangerous grounds. They become activated in exposure to a pathogen or exposure to some stimulus that would be able to bring those along. And they'll change configuration. And what we learned a very long time ago was that they upregulate a marker. It used to be called the peripheral benzodiazepine receptor, PBR. Later it became the translocator protein, TSP. And that gets upregulated, and that's our marker. And for a long time, people have been trying to image with this one there. The other thing we knew is that we could turn this on, that you could activate this. And the classic one is called LPS, lipopolysaccharide, also known as endotoxin, to be able to activate this. The thing that the chemists don't want in the tracer is what we're going to put in, first to an animal, and then to a human being, and see what we can do, see what kind of activation we can get of the brain's immune system, and can we measure that in vivo. We're going to use the particular tracer we're going to be using is a carbon-11 tracer, PBR28. This came out of Vic Plake and Bob Innes's lab. at NIMH some time ago. It was a second-generation marker, and it's got some interesting challenges there that we'll mention. A few years back, we first test-bedded this in monkeys. Great to have a nice monkey colony associated with your pet if you're going to be doing brain imaging. You can help develop the tracers, understand the physiology, understand the metabolites, figure out what you're going to get into. And if you get a black hole on a monkey scan, don't bother taking it to humans. This one, not so. So here we have this is our monkey MR, and we have a baseline scan that we do in the animal. Then we give endotoxin, and with carbon-11, as you all know, we can come back same day. And we did a scan at 1 hour post-endotoxin or at 4 hours post-endotoxin. And this was the measure, that same volume and distribution. We got a bit of an increase at 1 hour and a whopping increase at 4 hours. And that was good because we could also follow the peripheral effects, the changes in the cytokines that we could measure in plasma, and the peak there was about 3 to 4 hours. And you get these nice color images that look really good in publications because people believe it when it's in color for some reason. That was true a long time ago, maybe it's less true now. So the goal was, can we do this in human? Can we develop an experimental paradigm that can measure dynamic changes in microglial activation in the human brain using exactly the same paradigm? Okay? This could be used for a lot of things. It could be used as a stress test for your immune system in your brain. There's some really interesting ideas in Alzheimer's going on right now about how much the immune system might help to keep amyloid plaques at bay. And perhaps those people with an active immune system might hold off Alzheimer's a little longer. So I'm rooting for my immune system, really struggling. But it may just also be useful in drug development, being able to understand ways that you can modulate this. And it gets complicated because the immune response depends on whether it's the neuroprotective or neurodegenerative sides, and which sides are going to be relevant here. Okay? So here's our paradigm there. First, we have to do some genotyping. As I mentioned, we're using the second generation compound, DBR28, and what's been found really, really interesting, great PET finding, although I don't know what, is that there's a single nucleotide polymorphism for this site, and it affects the binding. It affects the affinity, and then you get high affinity binders, about 50%, medium affinity, about 40%, and low affinity binders, about 10%. Basically, you don't get a useful scan. So we genotyped first, we just did 8 subjects in the original publication. Okay, and so we're going to do a baseline scan. We're going to give them endotoxin. They're not real happy about that, but they get paid. And then we did a scan. We started about 3 hours post there to get that scan over 2 hour scan there. They're good by the next step. Okay? People will do a lot of things for money. It's remarkable. I don't know if that's true in Canada. It's certainly true in the States. So we measure our peripheral markers. We see how they go up and down. And we're going to do the same thing, all the same dynamic scans, arterial input functions, metabolites, etc. Here are the scans. We did the injected doses. Turned out to be a little lower in the afternoon. Nothing different. Specific activity. Masses were the same. Protein binding. These are, again, very low numbers, and there was no difference. 3% free fraction with some variability. Not too bad. We measured the cytokines that are going on just to validate, and we were seeing the increase in a variety of different IL-6, IL-8, etc. as a function of time, peaking at around the time of the challenge scan. Unlike the monkeys, we could ask the people how they felt, and so we could ask them about fatigue and various other measures, also peaking at about that time. This comes the money slide. How did we do? Here's our baseline scans. Here's our post-endotoxin scans. The other thing I learned in California is if you want to show numbers, you look in color. This is a really dramatic change there. 8 of 8 subjects all go up. Mean change there, it's about 40%. That's a big change 4 hours later in a PET study. Really, really large change there. And it happens everywhere. Pretty much every region. Not surprising, this is a mechanism that's ubiquitous in the brain. Now, reason why you would expect to see it in one place or another, but it's something that you've got just this extremely dramatic changes. And even when you separate out for the high binders and the mixed binders, you see that it affects it in both. This actually is a subtlety. If we put on my receptor binding hat, I'd say that the mixed binders have lower affinity. We were hoping to actually see a smaller change in them because they have a smaller specific signal. We didn't get it. The ends are too small to prove anything here, but it remains to be seen what's going on there. So this was pretty exciting. We think it's the first in humans to measure this dynamically in the human brain, and that the change in the brain went with cytokines and sickness symbols there. And 8 of 8, that's always good. It was 46% mean. And we think this might be really useful in both evaluating in disease states as well as looking at it in drug development. And we were very happy we got the notice from PNAS that this got accepted last week. And Christine San Diego presented this as a award at the Society of Nuclear Medicine meeting. And at the very end of that. So that was pretty exciting there. One more topic, and I think I saved the best for last. We're going to use a topic called SV2A. Anybody hear of that? Don't feel bad. I didn't hear about it either. This was one of those where we started working with a pharmaceutical company for their purposes to hit this target. SV2A we think is going to let us measure synapses, specifically synaptic density. And boy, wouldn't that be a good thing to measure. Obviously, you'd love to see it in every neurodegenerative disorder if you could measure the density of synapses. You'd like to see it in epilepsy, where there's great local changes in the epileptic focus. And then in diseases like schizophrenia, where there's huge changes in pruning there. Or even in depression, where there's great animal models showing spine sprouting and changes with depression. You name it, it would be fantastic. So how are we going to do this? We're going to do this with this SV2A. This is a marker. It's on synaptic vesicles. So here's a view of the presynaptic terminal. And on our synaptic vesicles, there's a lot of different proteins. One you're maybe more familiar with, one is called synaptophysin or synapsin. Those are classically used as in Western blotting to look for neurons and measure synaptic vesicles. SV2A is just another glycoprotein around on the side of those vesicles. It's not as common as synaptophysin. There's less per vesicle. But it's something that seems to be, based on literature, and there's not a lot of it, pretty ubiquitously expressed throughout the brain. Okay? Now this is an interesting target. It's beginning to be focused a bit. Okay? Here's a recent paper that came as a preclinical study that's tied to a clinical trial in Alzheimer's disease from Steve Strittmatter's lab at Yale, where here they're looking at a target, particular an agent, AZD0530, that's now in clinical trials, and they evaluated that first in mice models in the ABPPS1 mouse, and they use as their marker of synaptic density SV2A reactivity. They were able to show that the standard mice have reduced synaptic density, and in fact on their drug, in fact they showed it was recovered to normal. So that was pretty exciting that this might be a good in vivo measure of that. Now how did we get into this? Well, it turns out that SV2A is the target of the anti-epileptic drug Keppra, or levetiracetam. So if you know epilepsy, you know about Keppra. It's extraordinarily widely used. Okay? And it's a very simple molecule. Okay? It's pretty interesting there. And it was shown, actually, after its use was documented, it was shown that it was going to be about where a hit was at the SV2A. There's still a lot of controversy exactly what its mechanism of action is. What the company wants, and this is a company UCB, they wanted a faster acting version. Guess what? Kepra's going out of patent. I think it's already out. They have a new version, riveracetam. Sometimes that one's so hard to pronounce, I'd rather have a name with numbers. But in any case, they're trying to evaluate whether their drug gets in and hits the target quicker. And they wanted to use PET to be able to do that. So they collaborated with us to develop a compound, one in the series there. Here's the molecule that we've made. Has a name, they like to name it after themselves, so they call it UCB. This is J, so it's a 10th compound in this series. Not a bad molecule, not too difficult. We're labeling it with carbon 11. Those of you with quick eyes will notice the presence of 3 fluorines. So that's pretty handy for something you'd like to have something that would be labeled. It's one of those things when you're designing a molecule, it's nice when you don't know how fast the kinetics would be, can you measure it in the time course of the 20-minute half-life of carbon as opposed to the 2-hour half-life of fluorine. All right, first you start with the in vitro work, and you show the selectivity, very selective for SV2A over SV2B. SV2C is not present in the brain. You'd like to show, is it specific? Is it blockable by other targets? Here's the other agents that we use to say what % blockade did we get when we gave an agent specific for those other targets. Pretty much very selective for SV2 that we did there. Okay, you developed chemistry. This uses Suzuki cross-coupling. Very standard, very, very reliable kind of measurement there. Good yields, plenty of good specific activity, so something that we were very, very happy about. So we start with monkeys. We start with some anesthetized monkeys. What are we going to do? We're going to do this. We use a Focus 220 scanner that's from Siemens. And we do the same as we do with the human, arterial blood samples, dynamic scans. We did some test-retest. We did some blocking with levetiracetam. We also self-blocked, kind of like what we did with the glycine transporter, so that we can measure the in vivo KD, that same thing we did before. And here we're using the volume distribution by a graphical method, MA1. Here's what the images look like. It's not a very exciting image. It kind of looks like FDG, right? Well, it should, because it's going to neurons. It's going to be everywhere where there's a neuron, you're going to see it. So in terms of exciting novel distributions, we're not going to have it. But what we're looking at is not FDG. We're not looking at glucose metabolism. We're looking at the presence of this protein marker, SV2A, in the synaptic vesicles. We measure the metabolites. It metabolizes at a moderate speed, but we can handle. We looked at test, retest in different regions. Just a small n. Looked okay. Didn't look great. This was like the first time ever giving away the next step that the human was better than the monkey. So hold that thought. Here's the blocking agent. We gave the baseline and then give levetiracetam and do a blocking scan. 2 syntheses same day. This was with 65% blockade. Here's what the time activity curves look like. Time activity curves in different regions. All these are gray matter regions. I don't have the white matter curve. It's here, but it's below. Partial blockade. You see that the contrast tends to collapse. Or here's the same there. Probably this one's even better when we did the self-block. So here we're looking at the same animal, baseline, and different lower specific activities, progressively blocking, walking down the scatter plot, as I might say. And you see how the images are blocking here. We're showing those on an SUV scale. Here's where we do it numerically, where we're showing that beautiful dose-response curve. So everything lined up pretty well. We were getting reasonable, reliable measurements. We had the right dose-response curve. We were changing things ubiquitously in the gray matter. I didn't show you the white matter numbers. Feeling very comfortable. Is there, we would not expect any specific binding in the white matter, and if we do partial line correction, we think that agrees, but we really weren't that confident that we're selling that point too hard. Better to go to human. So we went to human. This is some material we showed at the S&M and the Brain PET conference recently. So we labeled that compound, and then we wanted to take it into humans. So we started with 5 healthy controls. Surprise, test, retest. Certainly be able to see how reliable this one would be. How good is this marker going to be? We started with a 2-hour scan on the HRT, and we did the blood and the whole 9 yards. I hope you've gotten that theme. Here's our measurements of the metabolites. I really like this slide because Short Finneman, who made that for me, did chromatography, and he did it in different colors, which was really perfect. So here's our parent compound and the metabolite. So we have 1 to 2 metabolites. Again, classically more polar in this HPLC conditions. We never really know what's going on with the metabolites. We don't ever absolutely know whether those are going into the brain. And it's one of those that we may eventually try to identify that molecule. We have a pretty good idea what it is and try to verify that, possibly with an animal study to verify. We have not done that yet. We have a lot of reasons why we don't think it's a problem. Here's the image. First image of this with UCBJ. Very high resolution. Very high selectivity there. Time courses of test and retest. Y-axis scale shows the SUV of 10. That's a good number. Really, really high uptake there into the brain. It's pretty exciting. We then did the modeling work. Everything worked. We tried the 1 tissue and 2 tissue model, and we get the same numbers. So a 1 tissue model works. And here's our volume of distribution numbers that we get in different cortical regions, lower value in white matter. This is reflecting, we hope, non-spliceable. We haven't proved it yet. Here's the uptake rate, K1. That's relating to blood flow. It's pretty good. Its number is about 0.3 and 0.4. That means this tracer is very well extracted. It means we're going to get a little blood flow information too. That'll be nice to be able to get both combinations there. But it's something that seems to work pretty well there. We looked at the test-retest reliability. I mean, I've showed you test-retest how many times today? This is my favorite slide in this whole talk. And you know why? Because look at these numbers. This is 3% test-retest variability. I've never seen that in 35 years. Okay? You're never going to see that kind of reliability, something that's working that well. We were just blown away by how good this would be. So the quality is so good, we can do pixel by pixel modeling. These are parametric images. Every pixel used with a 1 tissue model over time, like I talked about in the beginning, we're able to read out those volumes of distribution there. And this is going to be very important if we want to apply this in small regions of the brain, be able to get those test and retest. And they're really pretty gorgeous there. How long do we have to scan? We scan for 2 hours. We should check that. Well, it turns out if I scan only 60 minutes, I'm within under 5% agreement of what I got with 2 hours. That says I can do a short scan. That's pretty cool. Now, how about that reference region thing? We talked about that. I mean, clearly the white matter is very, very low, but is that going to be a good measure? Remember, like glycine transport, I found that the white matter and the gray matter didn't agree. Will I be able to use that here? We don't know that yet. We're going to know in the next couple months when we do our first Kepler study of a human being. But when we did use it, we still got very, very reliable results in our binding potential. Cerebellum up to 9%. That was our bad one. Again, 4-3%. We've begun to do patient study. I'll just show you one. Here's our first epilepsy subject. Classic temporal lobe epilepsy here. Here's the FDG scan, and I should add the disclaimer, this is done on a clinical PET scanner, so the resolution is much worse. But the ipsilateral contralateral contrast gives me 10% on FDG. It's getting 18% on the SV2A, just as an SUV image there. So it was suggestive that it was very, very good. So what was our take-home message? We're excited. We're trying to get NIH excited. Because we think we can measure synaptic density. And we'd like to know, what are we really learning? Are we really learning something new and cool and exciting? Or is this just going to be exactly the same as measuring gray matter density on an anatomical MR? We don't think so. But we've got to prove it. We've got to decide where we can do this, what areas it would be. And my God, you know, when we started talking about people, who would like to measure synaptic density in your patient population, we got a few responses. Basically, we're doing it in cocaine disorder and in schizophrenia and in depression, and we're starting in Parkinson's disease, and we're trying to do it in MS, and did I say Alzheimer's disease? Which would be pretty exciting. Because people have been using FDG as the surrogate marker for that, at least with PET, and it's not bad, but it's a little sensitive to things like glucose metabolism, like fed state, like neuronal activation. We don't think this is there. We've done a monkey study that is suggestive of that. We have way more to do with it. So we think it's a quantitative marker, and we'd love to be able to scan Alzheimer's disease and not have the need to fast. Just for that, that would be great. And of course, you remember that fluorine, those 3 fluorines on the molecules, and our chemists are working on the synthesis of the F18 version now. But the wide range of utility here, this could actually be important. And that's pretty cool. Okay, I've shown you lots of slides, lots of curves, lots of numbers, and lots of images. That's the important one. I probably don't have to tell this group that one. But that's the important image. Is your team, is your group. And that you can actually get them all facing in one direction at one time. That's actually really good. I mean, you guys are doing that really well right now, just to give you that credit for that. But it is just an amazing, it's so fun to have all the different cultures. And I don't mean French or English or Chinese or Japanese. I mean chemist or statistician or oncologist or psychologist or mathematician, whatever. And have everybody learn from each other about what may or may not be important in a given disease or a given disorder. I want to particularly acknowledge all the main people who've contributed specifically to the data that I've shown you today, show you to our sponsors, both from NIH and various foundations, etc., and oh yes, the pharmaceutical industry. We do that, and hopefully this is our logo here, where we have a little bit of our cyclotron particle accelerator that we came up with a long time ago, and we're pretty happy. And I'd like to thank you for your attention.
Thank you very much for this very interesting, very dense presentation. I'm sure that you have many questions on things you didn't have time to understand. So please, any questions?
On the PBR analog, what experiments have you done to control that you block for your PBR analog?
Right. So the block, in terms of the magnitude, some of the blocking studies were done with PK11195 to be able to demonstrate specific binding. In the monkey, I think we're seeing about 95% binding. In humans, this has been done, I think the Imanova group has done it. I think they've recently published to be able to demonstrate what fraction is specific binding. And they were showing that, if that's what your question would be, in the high affinity binders, about 2/3 of the binding was specific. In the medium, it was about 40% specific, which means that that 40% increase we got might be considered a 100% increase in the amount of the TSBO site may be activated microglia.
Right. I don't know if it would be the same thing in humans or in primates, but we're going to show next week at the WMIC that the creative inflammation in the brain causes vascular permeability. So you actually permeabilize the blood-brain barrier,
right,
which may, in principle, lead to non-specific accumulation of your compound.
So the thing and that's a great question, and it's very, very relevant for molecules that have trouble getting into the brain. This one doesn't. It's highly lipophilic. And we can quantitate that. We measure that net uptake. We measure K1s here on the order of also about 0.2 or 0.3, which suggests that with blood flow on the order of 0.6, we're 20 or 30% able to enter the blood-brain barrier in the normal brain. So we don't think that blood-brain barrier breakdown would affect that in any way, because we already have very good uptake there. It's a great question. It's really important when we have things that don't cross the blood-brain barrier relevant there. So the subtlety would be as if there's any specific, a differential mechanism that would be present when you have blood-brain barrier breakdown that would increase accumulation there or not. We haven't proven that there. It's certainly not something in these dynamic changes that we're seeing. I don't think if we're getting transient blood-brain barrier breakdown, we don't think that should be affecting it. It'd be a great one. It's something we could actually add a measurement to. A long time ago, we did use the rubidium in the brain to measure dynamic changes in blood-brain barrier breakdown. Obviously, MR, we can do that a little bit more directly. That was one where we actually did intracarotid injections of mannitol to open blood-brain barrier for direct delivery of chemotherapeutic agents to tumors. That was quite a tour de force too. So, but it's a great question, really relevant one.
Thank you for this presentation. We showed the utility of this marker for acute inflammation. But do you think that it can be used for chronic inflammation?
Great question.
Because the problem is in Alzheimer's that it starts 20 years before we get the whole picture. So how does it work?
So that's a great question. I mean, people have published that they can see that, and that they can see higher levels of this in people who have more advanced disease. Alzheimer's works a little bit simpler, perhaps, if there's it doesn't seem to be any inflammation going on in the cerebellum. So that makes it a little simpler measure without the arterial lines. Thankfully, that's an important tool there. I think the real worry is whether, it depends on your application. Are you going to ever get something that would be individually diagnostically useful? Probably not, okay? Whether we can learn that from a group, perhaps. The point I alluded to before, and we're having this really healthy discussion with our neurologists right now, is this good inflammation? Are we, how much of this is good or bad? Are we seeing, because you can look at Jaime Grutzendler at Yale has published some beautiful preclinical work showing how the microglia are wrapped around the plaque and preventing the growth of the plaque. So the question would be, how much are they activated, and what point are they activated? Are those the ones we're seeing in this image? So it's a matter of, this increase may be reflecting the fact that it's good, that it's keeping, like we said, keeping that at bay. I think that's going to be very exciting as we go forward. Do we want to facilitate this microglia response, or is this something that's actually going to be neurodegenerative? I think the jury is very far out on that issue. So it's a great question.
Okay.
You mentioned it in your presentation, but we tend to assume that the pharmacodynamic parameters remain the same aside from the affinity. And does anyone has ever tested systematically the effect of disease states such as inflammation, metabolic activity, systemic pH changes like in renal insufficiency, for example, on the different kinetic parameters using PET? Because I haven't seen yet a systematic study on these very basic questions.
Right. So, a couple things. Good question. So, this is where we claim that if, in the brain, the scanner gives us the brain data, and if we measure what's going on in the blood, we don't have to care about the rest of the body. So, renal insufficiency, liver cirrhosis may not matter. And in fact, we often will see that for the same injected dose or normalized to inject dose, you will see different clearance, different peripheral clearance. Clearly, whenever we do these blocking studies, very often we change the peripheral clearance. You change the availability of these sites throughout the body. And it actually gets disappointing because very often, so say you have a certain level and then you want to give a blocking dose and you hope your SUV goes down, but actually your blood level goes up so that it can often fully compensate for that. The real trick becomes then in what's going on in the brain, and can we take apart all those factors when you're in disease? What are those changes? Is this receptor? Is this an affinity change? Is this non-displaceable? Is the endogenous neurotransmitter changing? And that's what I didn't even mention. We lump all those together. We have an interesting result that we're trying to get to the bottom of now in cocaine dependence. We and the group from Toronto, am I allowed to use that word? Just checking.
Where's Toronto?
Okay, just checking. That we both found that in cocaine addicts that there seems to be higher binding in the substantia nigra, a nice small region source of dopamine, using the tracer PHNO. And PHNO is an interesting, challenging tracer because it binds both to D2 and D3. It's an agonist, but it has a much higher affinity for the D3. We've shown that in monkeys and blocking in humans. So now that becomes the question, is this a matter of, have they upregulated D3? Or is there just less dopamine binding? there? Is there less dopamine? We have a lot of evidence that there's reductions in dopamine synthesis, etc. So something there which is well concrete and well known, we still don't know. And we've been battling NIH to get us funding to try to do that. How would we do that? We would give a drug to deplete dopamine. We'd give alpha-methylparatyrosine. That goes in the category of fun days, a little worse than endotoxin in terms of the effects it does, because we're kind of making everybody a little Parkinsonian for a little while, to be able to deplete the dopamine and being able to measure how much we block and take that one apart. So that's one where we have a paradigm, we know how to do it, but it's hard, you know? And a part of this is a funding issue. And we've talked a little bit with some people today about getting to the bottom of some of these really interesting hard science issues to really feel good that we really are measuring what we claim. We can put up the glitzy stuff and we can get it published in PNAS, and that's exciting. Have we really gotten to the bottom of it? And all the rest of the work you need to do takes more funding than we often can get to do that. I wish that was easier. That would be better. We feel more comfortable in saying what we say.
You mentioned that on your wish list at the beginning of the talk would be an MRS PET sort of setup. And
I hear he's right. Yeah, I think he's working on it.
I'm sure he's working on it. Maybe think about following up on the metabolites from some of these tracers, and I guess that's part of your QC that you do, but you covered a lot of ground, so you probably didn't have a chance to get to it, but how do you approach that?
So it's so that's what I alluded to that, you know, we make the measurement in the blood, and we optimize an HPLC system to be able to do that fast, and we have, you know, carbon 11, if we have 5 or 6 points after the curve, we'll get that fractioned. And then we make an assumption in the brain, which might be okay, that the metabolites don't enter the brain. And we're wrong. We're always wrong. There's always going to be some. The question is some a lot or is some important? And that's going to depend a lot on how good is the uptake of our tracer. So for example, that SV2A, with that really high uptake, says it's getting in great, and even if those metabolites got in, it's probably a small fraction. But we do worry about it with compounds that have much, much lower activity. So, for example, a very common one that's challenging is the serotonin 1A compound, that WEI100635, which has beautiful contrast, had very, very low uptake in the cerebellum, and it turned out that probably half of that was a metabolite. An acid metabolite, one that was easily written in a paper, more polar, not likely to cross the blood-brain barrier. Okay? And then, of course, there's probably many of us in the room who actually choose to scan below the neck and found that there is interesting biology in clinical medicine in organs other than the brain. Shocking that that may be true. We have recently discovered that ourselves. And the shock of understanding what is it that we're measuring and how can we interpret it. And just talking about, you know, wanting a tracer that doesn't metabolize or does very, very little so that the signal is going to be easier to interpret. You know, there's the group, Simon Cherry's group is trying to build this whole body PET scanner, this Explorer. I don't know if you've heard about this, 2-meter long scanner, I'm sure right now. And they want to measure is whole body biodistribution, but it's going to be measuring whole body radioactivity distribution. And how much of that will be the tracer remains to be seen. So it's a great, great question. It's one that requires all of our cleverness. We can do it in chemistry, which would be best. We'd be happier with that. But we can also do it with the analysis on there and try to prove it. We need the animal data very, very tightly coupled to the human data to be able to interpret it, because very few people will let us remove their liver and even biopsy it to check for metabolites. It is interesting when we can do these studies tied to tracers. So for example, in this epilepsy, in the SV2A, I mentioned that we've begun to do an epilepsy. These patients are going to surgery, which means we're going to take out their tissue, and we can measure the SV2A and absolutely calibrate that against the PET signal. So that's a great luxury when you get to do that with human tissue. With cancer and biopsies, that's possible if the amount of tissue you get is reasonable. Great question. Really makes it one of our challenges, for sure.
Any more questions? No? If not, then let's thank you for this great talk.